Chris. Hi, students. Can I have a hi, Dennis, back?
Hi, Dennis.
Excellent. Thank you for working with me. In fact, I would argue that working together is the main thing that will make this work. The future of health will be digital. And not only digital, but it will also be a community effort. So in terms of health, we're really at the cusp of something absolutely amazing, where we will be able to diagnose diseases before symptoms really occur. We'll be able to ideally treat them or prevent them even from happening by going into the genome, for example, and making changes. And all of this will be because it has become digital, because we can analyze the huge amounts of data that our health represents in a way that really enables us to make changes early on before things get bad. But as I said, it's a community effort because it will require computational people in order to write code. It will require the community to demand that software will become open access, and it will require people to make the governments accountable to make this actually happening and going through to the healthcare system. So with that, let me tell you 3 stories. The first one is around cloud computing and what it actually is. The second one is how we can find disease genes. And the third one is around, well, now that we found disease genes, what can we do about them? And all of those, of course, have a cloud-based technology underpinning them that before was impossible to do, and now with this awesome compute power, we can do it. But before we go there, let me introduce CSIRO. So CSIRO is Australia's government research agency. At CSIRO, we're really passionate about translating research into products that people can use in their everyday lives. One of the most famous products is Wi-Fi, which is now used in 3 billion devices worldwide. We also developed the vaccine to Hendra virus, which is more deadly than Ebola. And on a lighter note, we developed the Total Wellbeing Diet book, which is now on the bestseller list alongside Harry Potter and the Da Vinci Code. So from that perspective, we have really a nice balance between products that people need and products that people enjoy. Now, I'm working for the eHealth Research Centre, which is the largest digital health initiative here in Australia. And its mission is to improve health through digital technologies and services. But we're worldwide quite unique in covering the full value chain, the full spectrum from basic science, where probably my research is more based on, to doing large-scale substantiation studies, and then to measuring what kind of impact we have had on the healthcare system. My group is the Transformational Bioinformatics group, where our charter is to bringing big data analysis to life science data. So specifically, we're focusing on the genome. Now, I know that Catherine North already told you much about the genome, but let me tell you about what I think the genome can do and why I'm really excited and passionate about it. So the genome, our DNA, really holds the blueprint for every single cell in our body, which means an enormous amount of information is stored in something as small as this tiny little genome. And in fact, it's not only affecting the way our disease genes, or what our disease risk is, what Catherine North talked about, but also the way we look. And there's something that you might have learned in your biology class that is called the hitchhiker's thumb. So the hitchhiker's thumb is the last digit of your thumb that is either really bendable or not. So in my case, it's not. But there will be 1 in 3 people in the audience where it's very bendable. And in fact, we developed a little app that makes this personal. So I encourage you to take out your mobile phone and scan in that QR code. That will get you to a web page. And on that web page, you can photograph your thumb, ideally with a relatively blank background. So hold it to the seat in front of you. And when you do that, it will give you a score. And if you're above 60, the angle of 60 degrees, congratulations, you're a hitchhiker. So you have a hitchhiker's thumb. as you can see on the picture here. So in fact, we can look at who has the most bendable thumb here in the audience by having a live tracker that records the pictures that were sent. But don't worry, they will be deleted after this. So when we go to this web page, we do have, so occasionally it does not work. So here is one, for example, 50%. Yeah. And up here we have a histogram of how many hitchhikers' thumbs there are. Now the interesting bit here is that, and I might go back to the presentation. So the interesting thing here is that it's a genomic trait. In fact, it's a recessive genomic trait. So both your parents must have been carriers for you to have a hitchhiker's thumb. And this original study was done in the 60s, where they were saying 1 in 3 people has this. But in fact, when you looked at the histogram, it's clear that actually it's not that common, right? The hitchhiker's thumb, the way that they defined it as being above 60% angle. So that means we need to do a little bit more research into that, into this trait. And that really represents, it's a physical trait, it's relatively easy to measure, yet we're still not quite sure what the distribution is, what the prevalence is, and what the inheritance pattern is on something as simple as that. Therefore, this really, I think, drives home that this whole area is very complex. And if you want to do something in the health space, in the disease space, it will be even more complicated than that. But ultimately, we will get there. Ultimately, we will understand how the genome translates into your future disease risks. For example, areas that we really well understand is Mendelian diseases, which Catherine North talked about. The classic example here is cystic fibrosis, where one change in the 3 billion letters of the genome can cause this devastating disease. With this information, with this much information encoded in the genome, it's not surprising that it will be used more and more in the clinical system. In fact, Frost and Sullivan estimates that by 2025, 50% of the world's population will have been sequenced. That produces more data in the genomic space than the typical big data disciplines, YouTube, astronomy, and Twitter combined. Let that sink in. I mean, this is an amazing amount of information, which on one hand is great because for the first time, we'll be able to analyze the amount of data that we need to build quantitative models and really understand the complexity of disease. On the other hand, it's quite daunting because no one really has done it before. No one really has analyzed that much data in one go before. And another analogy to drive this point home further is that usually people talk about it's astronomical when something is really, really big. I would argue that people should now say it's genomical, because when you think about it, the DNA is stretched out 2 meters long. There are on average 100 trillion cells in your body, and 100 trillion times 2 meters, that is larger than the universe. So in order to really understand your health and looking at every single cell in your body, you need to look at a space that is larger than the universe, and that is quite amazing. Which brings us to what is actually the cloud. So people jump straight to the conclusion that, well, in the cloud we can analyze all of this, because in the cloud we have virtually unlimited resources. And that's true to some extent. So public cloud providers, and here I list the top 5, Amazon, Azure, Alibaba, Google, and Oracle. But the way that I think about it is it's like an in-room entertainment system. So with an in-room entertainment system, you bring your iPad along or your iPhone. In that case, you still plug it in, but nowadays it's all Bluetooth. and you're using the speakers that are in the room in order to listen to your music. It's the same thing with the cloud. You can bring your software along and just plug it into the hardware that the public cloud providers offer. But not only that, so with an in-room system, you can add speakers that suit the type of music that you want to have. So for example, you need to add a subwoofer to add a bit of whoops, or have multiple speakers to add 3D sound. Same thing with the cloud. You can specify exactly what you need, whether it's a small machine or a large machine. You can even sprinkle some hardware, specialized hardware in there, for example, GPUs. And all of this you don't have to buy yourself, you can rent it in the cloud. And this is how I think about it. So with that, how can this now help us to find disease genes? So we are part of the international project MINE, which is one of the largest, if not the largest, single gene consortia that looks at a single disease consortia that looks at what is the molecular origin of a specific disease, in this case, ALS, which is a motor neuron disease that Stephen Hawking had. So with this prominence, clearly were the first ones to really have this done at an unprecedented scale. They have 22,000 individuals. Some of it have ALS and others are healthy, in order to really find what is the single or the multiple locations in the genome that drive this devastating disease. So therefore, clearly, it's groundbreaking research, and they need to use technology, the latest technology, in order to analyze this kind of data. So, how to do it? These are the genes. Every line here represents a genome, and every bar here, or every box, represents the changes or the mutations in the genome. So there are on average 2 million differences between you and the person sitting next to you, which makes you you, but also it can lead to disease. Most of those are harmless, but therefore the task is to find the ones that really are the culprits that drive the disease. So therefore, we're recruiting people that have the disease and comparing them to people that do not have the disease, with the aim of ideally finding the single location that stands out, that is enriched in the people with the disease and not there without the disease. Clearly, though, reality is a bit more complicated than that, especially complex multigenic diseases like ALS. Therefore, it will be multiple locations in the genome that work together in order to cause this disease. Resilience factors and amplification factors, which means that in order to find the set of genes that are driving this disease, we need to use something that is called machine learning, and more specifically, random forests. But ultimately, it's basically a big matrix that we want to compute over, where we have the individuals as rows and the differences between the individuals as columns, which for this kind of cohort, as I said, 22,000 individuals, averages out to about 80 million features. So 80 million times 22,000, that's 1.7 trillion data points to compute over. That is quite substantial. So, but luckily, there is a new digital technology that has come along that is called Spark. Let me walk you through what that means. So we're all familiar with the desktop compute, where the aim is to do small analyses ad hoc on our desktop machine. The step up from there is using high-performance compute clusters, where we have multiple CPUs on the nodes, and here this is what I'm representing as a node, with each box here representing a CPU. So therefore, with the high-performance compute clusters, you have more specialized hardware that have more CPUs on them, and you might have more nodes in a system. But the problem is that the computation is limited to the node boundary. So you can only compute within one node. And if you want to communicate between nodes in order to utilize more CPUs, you have to write bespoke code. And while you can do that, it takes forever to actually do it. Therefore, luckily, Spark has come along, which I think of it being dissolving the boundaries between nodes, and you can now use every single CPU in your cluster. So therefore, the task is, rather than compute intensive, which high-performance compute is focused on, it's now data intensive, what genomics is with 1.7 trillion data points. So we developed a machine learning library that is specifically catering for that. So machine learning has been around for a while, but usually not with 80 million features. Usually it's around 10,000 features of customer analysis data or things that are manually collected. With the genome, we're now 2 orders of magnitude larger than that. So therefore, there was no one that wrote a library like that, or a software that can deal with that kind of wide data. Therefore, in terms of accuracy and in terms of speed, Variant Spark is superior to all the things that we tested that are currently in the market. Therefore, the tagline is that it's faster because it can analyze 3,000 individuals with 80 million features, but it's also smarter than what is currently out there in that it now uses 80% less samples and still finds statistically significant signal. And that is relevant because every single can cost up to $1,000. Therefore, saving this much really makes a difference and makes some research possible in the first place. So therefore, we're quite fortunate to have cherished collaborators with Macquarie Uni and, of course, the Project Mine that I talked to you about earlier. So if you're interested in that, and stepping back here, not everyone is working in genomics, and you might not be working in genomics, but it's quite likely that you will be working with large data sets. So this could be log files, or it could be threat analysis, or it could be veteran sciences, or in VR. Therefore, with the datafication of everything, it will be so much easier to now collect information points per sample, which means the information per sample will grow. And this is exactly the space that we're sitting here in. Therefore, if you're interested in doing some sort of analysis with this, we have a starter script that gets you easily started with this. Who is actually computational here? Okay. Yes. In that case, my next question is a bit redundant in saying, are you familiar with notebooks? But I would argue that going forward, it will be, because it's so much easy, or it's really easy to now analyze data with a computer, that you probably will learn about that in university. You probably will be required to analyze some data and get insights from your data. Therefore, a notebook-style analysis is really nice because it's basically a recipe book where you have the recipe written up already, and all you need to do is execute that recipe like cooking a meal, and out comes a picture, like a juicy, nice, yummy picture at the end. And that's basically what I'm showing here as on the notebook, where you have the bit of code here that gets executed and out comes a figure. So we do have a starter script that does that, and a nice little data set that comes along with it, which is our hipster data set. So the hipster data set represents what a complex genetic disease is, with the hipster as our phenotype, our disease. So if you're not familiar with what a hipster is, a hipster here, at least in Australia, is defined as a Swiss millennial, I guess, that drinks lots of coffee and has checked shirts. So therefore, checked shirtness and drinking coffee is actually a genetic trait, or can be defined as a genetic trait. And from there, we build our disease data set, our hipster disease data set. Anyway, the point is that going forward, it will be so easy to analyze data that everyone will be doing it, from data-driven journalists who are already doing it to actual researchers looking at their data set. And one way of doing it is through databricks. So databricks is a startup company in the US, one of the unicorns in the US that has grown from virtually zero to exponentially a huge amount of people working now for them, which develops exactly those notebooks. And what is the easy thing about it is that you just go to your notebook and it automatically links up with the cloud providers like AWS and Azure. So you don't have to do anything about spinning up the infrastructure itself, like hooking up the speakers to your iPad analogy. You don't have to do that. Databricks is doing it for you. All you do is run that bit of code and get the juicy picture out of it. If Google Cloud Platforms is your thing, then Terra is the sort of equivalent to that, in that they provide a notebook and you can link up with the analysis tool behind it on Google. If you want to be a bit more computational, and for the 2 people in the audience, that might be your thing, then diving deeper into the code and spinning up the resources yourself is easy with Marketplace. So Marketplace is basically an app store on your iPhone where you can select certain tasks, certain apps, and they're provided by third parties, like, for example, us. So therefore, we have an app in the App Store, in the Marketplace, that you can just install, click on it, and it will automatically spin up the hardware for you. So let's come back to the actual science. Finding disease genes was our task, especially in the ALS space. So we and others have identified a particular gene called SOD1 to be responsible for a certain type of familial ALS disease. And what was shown is that silencing these genes, like knocking that gene out so that it's not expressed anymore, causes the disease to slow down because it stops the death of neurons, at least in mice models. But ultimately, this is a potential pathway towards not a cure, but at least a treatment to slow down the progression of ALS. But the question is, how do you knock out a gene in a living organism? And here is where we come to our third story, which is around genome editing. So therefore, you might have heard of CRISPR, which is this revolutionizing technology that allows you to edit the genome of living cells. And it does that by going to a specific location in the genome, interacting with that genome, of either cutting there and letting it repair, or inserting a new bit of DNA into a specific location in the genome. So therefore, ultimately, this will allow new treatment areas where you can deliver this machinery, this CRISPR machinery, to the specific tissue that you want to edit. In our case, that will be neuronal tissues. And not only that, you then go to a specific location in those cells in order to deliver, correct the mutation that caused the problem in the first place. And when you think about this, this is not only relevant for the ALS space, but it's basic for every kind of hereditary or genetic disease you can think of, but also for a lot of other diseases like cancer, where this is now being trialed. So it really will change how we think about treating individuals and how medicine will be practiced in the future. But at this stage, it's still quite tricky to actually do that, to go to a specific location in the 3 billion letters of the genome and make the change. And why is that? The problem here is that it's like finding a grain of sand on a beach. It has to be the right shape, the right size, and the right color for the machinery to interact with it. But then you're not done. Like, imagine you went through the beach and picked up all the candidates in your little bucket. But then you're not done, because you also need to make sure that this grain of sand that you have as a potential candidate for interacting with the genome is unique in the rest of the genome, so that the CRISPR machinery is not accidentally binding over there and causing more harm than good. Therefore, with your little bucket of sand, you now have to go through the whole beach again and make sure that it is sufficiently unique from everything else on the beach in order to say, yep, this particular target site, this particular grain of sand is the one that I want to choose. And that is quite a computational, expensive task. So we developed GT-scan, which is Genomic Target Scan, that goes through the genome and looks for those target sites. So every bar here represents this grain of sand, this location in the genome that could be interacting with the DNA. And in color, we represent the ones that are actually good, the ones that are unique and have a high affinity to the genome. And as you can see, they're quite close to each other. A researcher would not be able to go in there and would be able to eyeball the difference between a good and a bad target site. And if this is an application where you want to edit human tissue, precious human tissue, you better not get it wrong. Therefore, computationally optimizing the approach is the way, at least we think, is the way forward for actually achieving that and bringing it into the clinical practice. So, as I said, this is quite computationally expensive for a web app to actually do this. But luckily, again, a new technology, a new cloud-based technology has come along in order to help us with that. So again, let me walk you through that. Starting off with our desktop computer. So with the desktop computer, the task is that you have full flexibility and it's cost effective. Like on your computer, you can install whatever you want and you can optimize it to make it really cheap. And the equivalent is owning your own car. You can go wherever you want and you can do whatever you want to with that car. Problem though is that there is a huge overhead with that. You have to look after the car, you have to bring it to service in order for it to run. And if you don't want to do that, you can rent a car. So the equivalent to that is renting a cloud service, as we talked about earlier. Problem though is that it's not very scalable. Like imagine you're going to IKEA and you pick up a new couch. Yes, you have rented that car, but if that car is not able to carry a couch, you can't just click your finger and convert it into a truck. However, you could do that with a chauffeur, right? You could have the chauffeur bring you to IKEA, you buy the couch, and then you send that chauffeur home to come back with a pickup truck. Therefore, this is the auto-scaling group or the auto-scaling capability in the cloud. Problem though with that is really expensive. It's like a chauffeur. No normal person probably would be able to afford a chauffeur for a shopping trip to IKEA. They probably wouldn't go to IKEA anyway. So therefore, this is where serverless comes in. Serverless provides all the opportunities that you would have from a chauffeur, except that it's now affordable. It's scalable, it's flexible, and you can do whatever you want with it. So therefore, the equivalent that I think of it, it's like Uber, it's like a ride-sharing app where you can specify a certain type of car that you want to have at a certain location, then and there, with no overhead because there are so many cars around it. And I think this is the unique thing about it, that you can specify whatever you want instantaneously, you can get it in terms of the compute. So it's like recruiting CPUs when and where you need it. So we were one of the first research groups to actually use this new technology to do something real with it for the real world in terms of research. So GT-scan, the Genomic Target Scan application, the search engine for the genome, we implemented with serverless technology. And I'm not going to go through the architecture. If you're the two people that are interested, that are computational, if you're interested in this, I encourage you to go to YouTube, where we have a "This is my architecture" video that goes into detail of how we actually did that. But I think what I want you to go away with this is this figure in terms of the costs. So if we were to implementing it in the traditional way on servers, we would have to fork out $714 per month in order to provide this service to the research community, of having a web service running 24/7 that can scale from a single gene to the whole genome if that researcher wanted to. Which, for a research organization is really costly. But with serverless, we can bring it down to $30 a month, which is affordable. Now, with the benefit of serverless is that it's modular. So you can exchange certain elements to it and play around with it. And therefore, if you do that, you can bring the cost down even further. So now it's just $2.50. Like, it costs less than a coffee per month for us to offer this service, which really democratizes the access to high-quality scoring of target sites in the genome. And I think this really brings home how new technology makes things that before would have been prohibitively expensive and therefore impossible to do, now makes them possible to think. And this is, I think this is really the take-home message here, is that new technologies come out all the time that make things that were impossible just yesterday now possible, and therefore the sky is the limit for dreamers. Now, DTScan is available not only on AWS, it has started there, but now it's available on other cloud providers, for example, Alibaba and Azure. And I do have an overview here, if you're getting the slides later on. I do have an overview here of the different technologies that are in the different providers. Yes, there are different cloud providers, but conceptually, they have the same services, if you want, available in there. So therefore, I truly think that once you go serverless, you never go back, because it will allow you to innovate really cheaply. It allows you to develop something that you can test on a small scale, which costs you nothing, and it instantaneously, without you having to do anything special, scale up to production, scale up to the real world, to a real world application. So therefore, innovation, I think, becomes affordable because you suddenly can exchange certain elements in your workflow without having to reinvent the whole thing with a new platform. Therefore, it's not surprising to me, at least, that it's estimated that by 2025, this market, the serverless market, will be a $20 billion market worldwide. So, as I said, my research team is focused on using big data analysis tools in order to advance life science applications. We talked about finding disease genes where VariantSpark is the technology, the machine learning technology, in order to analyze high-dimensional data. And we talked about DTScan, the search engine for the genome, where a researcher can jump on this web service, type in the gene that they want to edit, and it comes up with a ranked list of potential candidates, potential grains of sand on the beach that people should focus on. But ultimately, we want to treat people. Therefore, we do have a new technology just developed, and at this stage, I probably don't have to tell you that it's a serverless technology as well. That is where the aim is to combine genomic data with clinical data in order to make personalized decisions. However, I don't have anything to show you on that, so therefore I encourage you to, again, scan in that QR code. The thing before was a test. Scan in the QR code again. That will get you to a web page, and there, our web page. And there we have a blog post where we, every week, publish a new article around our research. But if you're now thinking that this is actually quite interesting, and maybe I do want to skill up a little bit more on cloud, there are ways of doing that now easily and in a fun way, specifically for students. Like all the 4 main cloud providers, they have credits, where as a student you can sign up, you get cloud credits for free that lets you play online, that lets you play in the cloud and test out certain technology there. It also gives you access to free courses, and in the end it can give you a accreditation where you can put that on your CV and everyone knows that you're accredited for cloud, which I think going forward will be a huge benefit having that on your CV. Good, so with that, the 3 things to remember are the datification of everything, like sensors and everything that has come in, means that more and more data points will be collected about the individual sample, meaning that the data sets will grow. The number of rows that describe this particular sample will grow in every domain, not just genomics. And currently, the technologies, especially the machine learning technologies, are not capable of dealing with that. Genomics was the first discipline to really have that problem. Therefore, we were the first to develop something in that space. So therefore, if you come across a data set that is extremely high dimensional, that is extremely wide, consider variance path as one analysis methodology. Also, serverless architecture, it really will enable the future. It will enable rapid prototyping and bringing your ideas, making your ideas, I think, a reality. With serverless technology, it will be so much easier to actually do that, to quickly put together a wireframe prototype in order to show people what you're thinking of. And then that wireframe product can easily be scaled out to a real-world product that cater for a much larger traffic or much larger data sets. If you're in the genomic space, in the genome engineering space, which in my mind will be where the future is in terms of medical, life science, biosecurity, manufacturing applications, everything, I guess, can be to some extent influenced or improved by genome engineering, by CRISPR technologies. So if you're in that space, consider GT-Scan as one of the technologies to really democratizing the access to high-quality analysis to prioritize how you edit the genome with sensitivity and specificity. But I think the most important take-home message is that bioinformatics really has become a team sport. Right? The solutions that were originally developed as a single software package, we're well past that. Everything that we now develop depends on other things. Therefore, going forward, we'll be building on each other's solutions. And therefore, cloud will enable that. Because you might build on a technology that is already up and running in the cloud, and all you do is bring on your brilliant idea in order to improve it, in order to make it better. So the solutions that we will be building together in the future will be larger than the sum of its parts. So with that, let me thank the team, and are there questions?